Good morning. This is going to be our first lecture on Bio 211/201, Introductory Genetics and Cell Physiology, also known as Genetics 1. Our first topic for discussion this morning is the foundation of genetics and the works of Gregor Mendel. Before I begin the lecture, I have some learning outcomes which will guide you in the course of this lecture. At the end of this topic in this module, you are expected to know the following: where to define genetics and know its importance to life, describe the various branches of genetics, have a basic understanding of the foundation of classical genetics, develop an understanding of the principle of inheritance, understand the genetic basis of life processes, define various genetic terms such as pure lines, genotype, phenotype, filial generation, dominant trait, recessive traits, monohybrid cross, dihybrid cross, homozygote, heterozygote, backcross, and test cross. Know how to draw and explain genetic diagrams. Understand some of the methods used in genetic study of living organisms, as well as develop an ability to use genetic principles in solving appropriate biological problems. Having said that, what then is genetics? Simply put, it's a biological science that deals with the mechanism of heredity and the causes of variation in living organisms. Why that siblings of the same parents do not all look alike, but some have some facial appearance or resemblance to either of the parents. Sometimes you are told that you look more like your dad, or you have the nose, facial look of your dad or your mom. Genetics enables us to study this because genes are responsible for these traits. It explains the mechanism and the basis for both similarities and differences between related individuals. As far back as 1906, Bartlett coined the name genetics for the study of the physiology of heredity and variation. And it was derived from the Greek word "rotgen," which means to become or to grow into. There are different branches of genetics. These include plant genetics, which covers the area of Gregor Mendel, genetics of plants, passing of traits from one generation to the other, animal genetics. Human genetics deals with the study of heredity of human traits, genetic disorders, and correction of such disorders. Microbial genetics deals with the genetics of microorganisms such as the bacteria. Mycogenetics deals with genetics of fungi, how fungi reproduce, how they pass on genetic traits from one generation to the other. Mendelian genetics, which is what we're going to look at in this first topic of this lecture, which deals with the study of heredity of both qualitative and quantitative traits and the influence of the environment on their expression. Quantitative genetics deals with the study of the heredity of quantitative traits, which are traits that are measurable, such as height, weight, human milk production in cattle, as well as grain yield in crops. Molecular genetics deals with the study of the structure and function of the gene and the regulation of its activity. We also have transmission genetics that deals with the study of the mode of transmission of genes from one generation to another. Clinical genetics that deals with the detection of hereditary diseases in man, thereby suggesting possible cure for such diseases such as hemophilia, phenylketonuria. Immunogenetics that deals with the genetics of production of different antibodies. We also have behavioral genetics that deals with the interaction of genes with the environment to build a particular pattern of behavior. For example, you have identical twins, and you take one of them and place them in somewhere like Ojo Elegba in Lagos, and you take the other one to probably Washington, D.C., or live in the White House, after 10 years, bring them together, their behavior will not be the same because it will be influenced by the environment in which they grow up. Behavioral genetics studies that. Biochemical genetics gives the biochemical explanation of various genetic phenomena, while population genetics deals with the genetics of the different population of living organisms, plants and animals. The genetics that deals with genetic knowledge that deals with the development of biology of organisms is known as developmental genetics. How the organism changes from one stage to the other in its growth cycle. Ecological genetics deals with the genetics of ecological phenomena. Look at the historical background of genetics. Before the time of Gregor Mendel, there were several hypotheses that were brought about by various individuals, scientists, philosophers, who tried to explain inheritance. There are basically 5 of them. The first is the vapor and fluid theory by early Greek philosophers such as Pythagoras and Aristotle. For Pythagoras, he proposed the theory that every organ of the male animal gives out vapor which unite and form the embryo in the uterus of the female. That's quite funny. That was his own idea, but of course we know that that is not true. Aristotle, another Greek philosopher, proposed that the semen of man has a vitalizing effect which considers purified blood. Of course, he had a point here. The semen has life, but definitely it's not purified blood. He believed that the female furnished the inert building material, while the male gives motion and new life to the material. Well, that innate building material we now know is the egg, which is being fertilized by the sperm, which is the fertilizing effect. He also thought that a giraffe was a hybrid animal, the result of a cross between a leopard and a camel. Although he had some idea about inheritance, but he did not actually get it right. We also have the preformation theory. Leonardo da Vinci, the famous painter, proposed that the male and female parents contributed equally to the heredity of the offspring. He was right on point. Because by the time we look at Mendelian genetics, you notice that you have 50-50 contribution of gametes from both parents. W. Harvey speculated that living things originated from eggs and that the semen only plays a vitalizing role. He also had a point because the egg is fertilized by the semen, the sperm from the male with a vitalizing effect, and after fertilization it grows into an embryo. But this does not occur in all living things. The graph introduced by parental inheritance, when it suggested that both parents contribute to the heredity because he observed that the progeny would possess the characteristics of both parents. Talk about a blending inheritance. Some traits are taken from one parent, while some traits are taken from the other parent. Some other introduced the preformation theory, which advanced the concept of preformed embryo in the sex cells, which is known as homunculus. According to him, in the gametes of the male and female, you already have a preformed embryo known as homunculus. Of course, this is not right. He was wrong. Looking at the epigenetic theory, CFO proposed that neither the egg nor the sperm had a structure of homunculus, a preformed embryo. He negated the study of the previous researcher that says that the gametes contain preformed embryo. Rather, he went on to say that the gametes contain undifferentiated living substance, which was capable of forming the organized body after fertilization, thereby refuting the preformation theory. Of course, his observation, his theory is in the right direction. Because when the egg and the sperm mix during sexual intercourse or coitus, fertilization takes place and a new individual is developed and formed. The particularly curious one is the theory of pangenesis. Maupertuis refuted the preformation theory when he proposed the concept of biparental inheritance by elementary particles. According to him, both parents produce the semen, which contain many tiny particles. And the semen of both parents unite during sexual intercourse to form the embryo. He had an idea, but of course he was wrong. Semen is always produced by the male and not the female, and does not contain tiny particles. In 1868, Charles Darwin proposed the theory of pangenesis, which depends on the particulate theory that has acquired and inherited. The pangenesis was an idea that males and females formed pan genes in every organ, not just reproductive organ, but in all organs of the body, including the somatic cells and the gametic cells. And these pan genes subsequently move through their blood to the genitals and then to the children. Of course, he was wrong, as that's not accurate. Pangenesis is that each part of the animal body produces minute particles called germs, which are first collected in the blood and later constrained in reproductive organs. But this was disproved by Francis Galton and Charles Darwin's cousin. The last theory was that of the germplasm theory, which was postulated by Westphal. And it states that the body of organisms contains somatic cells and reproductive cells, which is correct. But he called reproductive cells germplasm and the body cells somatoplasm. But now we know that the body cells are somatic cells and reproductive cells are gametic cells. The germplasm gives life from one generation to another. According to him, germplasm can form somatoplasm, but somatoplasm cannot form germplasm, which is absolutely correct. Gametic cells, when they fuse, can form body cells in a new offspring. But the body cells, the somatic cells, can never form gametic cells. So he was right in that theory. Hence, the changes in the structure of somatic cells, which are caused by the environment, cannot influence the reproductive cells. For example, if you cut the tail of a mouse, it does not mean that when it gives birth to its offspring, those offspring will not have a tail. Thus, this refuted the Jean-Baptiste Lamarck's theory of inheritance of acquired traits. Now, let's look at Gregor Mendel and classical genetics. Gregor Mendel is regarded as the father of genetics because of his work on the garden pea, Pisum sativum, for a period of 7 years, 7 to 8 years. He was an Austrian monk and carried out genetic crosses in the garden of the monastery with over 28,000 pea plants. And he reported his work in a meeting of the Association of Natural Research in Brno in 1865 and formulated two fundamental laws of inheritance, which has given us a good insight into inheritance in living organisms. Now let's take a look at Mendel's first law of inheritance, known as the law of segregation. At the time of Mendel, the word gene was not yet being used, so he called them hereditable factors. And it says that these factors occur in pairs that can be separated in the process of sexual reproduction. And the two members of a gene pair, also known as allele, segregate from each other in the formation of gametes. If you look at gametogenesis, the production of gametes, you have oogenesis and spermatogenesis. And during these stages of gamete formation, the diploid cell, after the second meiotic division, becomes haploid. Now, what he's saying is that the particular gene pair has an allele, which are alternative forms of the gene. For example, we have a gene for height, it will be tall or short. Which of these gene pair will move into the gamete occurs by chance as they segregate. They will both not go into the new cell that has been formed, but one of each of the pair will move to a different cell. When Mendel carried out the experiment, he developed pure lines. That means that he used true to type P plants that had 7 discrete characters or contrasting characters. For example, height, tall or short, pod color, yellow or green, flower color, purple or white. And he made sure that after selfing from one generation to the other, they all bred true for those traits. And he counted his results mathematically and kept statistical notes, which helped him to draw conclusions to his experiments. These are some of the contrasting traits he went through. Some are dominant, while the others are recessive. For seed shape, it could be round, spherical or dented. The seed color could be yellow or green. The flower could be purple or white. The pod shape could be inflated or constricted. The pod color could be green or yellow. And the plant could be tall or short. How did he carry out this cross? To cross two different pea plants, Mendel used an artistic brush. What he did, the carpel is the female part of the plant. He removed the stamen, which contains the pollen grains, leaving only the carpel that contains the ovule, the female part. And then he transferred the pollen grains from the stamen of the white flower to the carpel of the purple flower. It's called cross-pollination. The pollinated carpel, when matured, produces a pod and gives rise to seeds. These seeds are harvested and grown out in the field to give the first filial generation. Because purple is dominant to white, during the first filial generation of the offspring, all the plants appear with purple flowers. So looking at the first law of Mendel, known as law of segregation, it states that heritable factors occur in pairs that can be separated in the process of sexual reproduction. And each parent contributes one and only one allele or part of a gene pair to an egg or sperm. As seen in the diagram here, this is parent 1 with capital S, let's say for smooth seeds, and this is parent 2 with small s for wrinkled seeds. They produce only one gamete out of these two pairs. It's segregation. It segregates to get just one out of the gene pair. Same thing with the other parent. And when there's fertilization, we now have a diploid cell being tall, being smooth, a smooth seed in F filial generation. When this first F filial generation segregation takes place, it can have either of these gametes, either the capital S or the small s. When it is solved using a Punnett square, we're going to have three smooth seeds and one wrinkled seed. Now, it's interesting to note that in the work of Gregor Mendel, he carried out two different types of experiments. For him to obtain the first law of inheritance, he carried out a monohybrid cross experiment involving a pair of contrasting characters. And he crossed pure breeding smooth-seeded plants or yellow-seeded plants with a variety that had always produced wrinkled seeds or green seeds. And observed at the end of the day, not a blending giving a different color, but at the F1 all produced smooth yellow seeds. The following year, he allowed the smooth seeds to self-fertilize and obtain something different. He recovered about 7,324 seeds that are smooth and 7,324 seeds, out of which 5,474 were smooth and 1,850 were wrinkled. Don't forget that smooth is dominant to wrinkled, and it was in ratio 3:1 in the second filial generation. Now, when making genetic crosses, take note of the symbols you use. For the parental generation, it's given as P1 generation, and you show the parent, male and female. And after fertilization, the offspring obtained from the P1 generation is known as the first filial generation. And after the first filial generation, you have possible gametes that are obtained, and selfing of those possible gametes gives the second filial generation. Now, assuming you have a cross between a smooth and wrinkled, that's the parental generation. In the F1, all of them will have smooth seeds. And after the F2, you have 5,474 smooth and 1,850 wrinkled, which gives a ratio of 3:1. Now, it's interesting to note that meiosis, which was not known during the time of Gregor Mendel, explains how the traits are inherited. Because you have segregation taking place, separation of the gametes, especially at the metaphase, when the chromosome, homologous chromosome line of the spindle fiber, they segregate and assort themselves out independently. The F1 offspring showed only two of the two parental traits, and always the same trait. And the results were always the same regardless of which parent donated the pollen. The trait not shown in the F1 reappeared in the F2 in about 25% of the offspring. And this is regarded as the recessive trait. Traits remained unchanged when passed to offspring. They did not blend in any offspring but behaved as separate units. So there is no blended inheritance here. When he also made reciprocal crosses, they showed that each parent made an equal contribution to the offspring, 50-50 contribution to the offspring. So from his first experiment, Mendel had some conclusions. Evidence indicated that hereditary factors could be hidden or unexpressed. Those factors that are hidden in the first filial generation are known as the recessive traits. And the organism with alternate forms of a gene will express the form that is dominant. That is the dominant trait. It will always show itself in the first filial generation. As long as it is present, it will appear in the first filial generation. The term phenotype refers to the outward appearance of a trait. what you see, facial, facial appearance or physical appearance of the trait. While the term genotype was used to describe the genetic makeup of the organism. He also concluded that male and female contributed equally to the offspring's genetic makeup. Therefore, the number of traits was probably 2 traits. He also used uppercase letters to denote dominant traits and lowercase letters for recessive traits. Take note of this, especially when you are drawing your genetic diagrams or making genetic crosses. Let's take a look at the second law of Gregor Mendel, which is law of independent assortment. This was his second experiment, and this involved a dihybrid cross experiment, not the monohybrid cross that gave rise to the first law of inheritance, but a dihybrid cross, a cross involving a pair of 2 contrasting characters. Now, this law states that the pairs of alleles needed for each trait are passed on to the offspring independently of one another, and this occurs in meiosis II and metaphase when they line up in the equator of the spindle fiber and move for opposite poles independent of one another. So it means that the offspring can possess combination of genes that neither parent possess. So a second law of independent assortment is also a consequence of meiosis in a dihybrid cross. As I earlier said, Mendel started with true breeding plants that had smooth yellow seeds and crossed them with true breeding plants having green wrinkled seeds. All seeds in F1 have smooth yellow seeds, and in F2 it had 315 smooth yellow seeds, 108 smooth greens, 101 wrinkled yellow, and 32 wrinkled green, which is approximately 9331. When he analyzed each trait separately, he also gave a 3:1 ratio. Like we said, in the first law of inheritance, there is a segregation of the S, capital S for smooth and small s for wrinkled allele, and also a segregation of capital Y for yellow and small y for green allele. If you take a good look at the diagram here, we have the diploid parent, and this is the genotype. It is smooth and yellow, because capital S stands for smooth and capital Y stands for yellow, and they are both dominant also over small s, which is wrinkled, and small y, which is green. And you have the chromosomes containing these different alleles. So during meiosis I, homologous chromosomes line up and are separated into new cells. That's where segregation takes place. And at the end of meiosis II, you have 4 daughter cells containing 4 possible gametes: smooth and yellow, wrinkled and green, smooth and green, and wrinkled and yellow. According to the second law of Gregor Mendel, the capital S assorts with either capital Y or small y, and the small s assorts independently with either capital Y or small y. Now, suppose a pair of alleles control seed texture, with capital S containing smooth, while small s controls wrinkled, and smooth is dominant over wrinkled. Another pair of alleles controls seed color, with capital Y controlling yellow seed, while small y controls green seed. Yellow is dominant over green. Therefore, if you have your parental generation with smooth and yellow seeds and the other parental generation with wrinkled and green seeds, they all segregate and give one gamete, smooth and yellow, wrinkled and green. And when there's fertilization, at the end of the day, the F1 will be all smooth and yellow. Now, looking at the F1 genotype, we can get 4 possible gametes from here: smooth and yellow, smooth and green, wrinkled and yellow, and wrinkled and green. And when these gametes are allowed to fertilize in the F2 generation, you're going to have a difference. Now, the chance of any gamete having a capital Y is 1 over 2. Because either capital Y or small y, round, yellow, or green. And chance of any one gamete having capital S is 1 over 2. It's either smooth or wrinkled. Therefore, the chance of a gamete having both yellow and smooth is therefore given by the product of their individual chances, which is 1 over 2 times 1 over 2, giving 1 over 4. And the chance of a gamete having both one S is the product of their individual chances. And the chance of these gametes forming a genotype is given by 1 over 4 by 1 over 4. Simply because you have 1 over 4 for yellow and smooth, and 1 over 4 for wrinkled and green. So for the genotype to become together, you have 1 over 4 times 1 over 4, which is 1 over 16. That's going to give us 16 different combinations of gametes, of genotypes, as seen in the Punnett square. Since there are more possible combinations to produce a smooth yellow phenotype, it could be either of any of these 4 different genotypes: homozygous smooth yellow and heterozygous smooth yellow, as the case may be, as we see in the Punnett square. You have the male gamete and the female gamete, smooth and yellow, smooth and green, wrinkled yellow, wrinkled green. So in obtaining the genotypes in the 16 boxes, you multiply each allele, each gamete, with the gamete of the male. And this with the gamete of the male to get this with the gamete of the male to get this, and this with the gamete of the male to get this. And this gives the segregation ratio of 9:3:3:1. Now, let's have a short exercise. Can you identify the various phenotypes of the F2 progeny given in the above Punnett square? What is the proportion of smooth and yellow, smooth and green, wrinkled and green, wrinkled and yellow, and wrinkled and green? Find out the proportion and identify the genotypes, the phenotypes. Because the inheritance of flower color has no effect on inheritance of seed color, a white flowered plant that produces green peas can come from a white flowered parent that produces yellow peas, because they assort themselves independently. Remember that Mendel considered 2 traits per cross: the hybrid as opposed to single trait crosses, monohybrid. And the resulting F2 generation did not have 3:1 dominant to recessive phenotypic ratio, but 9:3:3:1. Assuming that we have 2 loci, a loci are positions of the gene on chromosomes, on 2 chromosomes, round or wrinkled seed, and another loci for yellow and green seed. These alleles will sort themselves out independently so that they can separate or go together at anaphase 1 or 2 of meiosis. So if you have a cross of round and yellow with wrinkled and green, it produces round and yellow in the F1 generation. This round and yellow will produce 4 possible types of gametes, which is round and yellow, round and green, wrinkled and yellow, and wrinkled and green. I can combine in 16 ways to produce 9 genotypes, 4 phenotypes in the F2 generation, as you see in the Punnett square. You have 9 round and yellow, 1, 2, 3, 4, 5, 6, 7, 8, 9. They are all round and yellow because you have capital arrow and capital Y in the genotype. We also have round and green, 1, 2, 3. It is round because of capital arrow, capital arrow, capital arrow, which is dominant over small arrow. And it is green because you have small y, small y, small y. Then we have 3 wrinkled and green, 1, 2, 3. It is wrinkled and green because they are all small letters. Sorry, wrinkled and yellow. Wrinkled because they are all small letters. Arrow, small arrow, small arrow. But this is yellow because you have a capital Y there. Capital Y is dominant over small y. So these 3 are wrinkled and yellow, but 1 wrinkled and green, giving a 9-3-3-1 phenotypic ratio. Based on the 2 laws of inheritance, you can also talk about the law of dominance, which says that hybrid offspring will only inherit the dominant trait in the phenotype. For example, you have your F1 generation, the dominant traits that will appear in that phenotype. The alleles that are suppressed in that first generation are called recessive traits, while the alleles that determine the traits are known as the dominant traits, not dominant traits. Now there are some links here for further study on your own to find out about Gregor Mendel, Mendel's papers. You can go to that link and read more about Gregor Mendel's experiments. Now, apart from making crosses, you can also test to find out the type of genotypes you have in a dihybrid cross. And this can be done in 2 ways, either by selfing or making a test cross. Now, in selfing, an individual that is heterozygous will segregate when selfed with regard to the locus where it is heterozygous. Assume, for example, an individual that has smooth yellow seeds. If the individual is of the genotype capital S, small s, capital Y, small s, capital Y, then we have 2 possible gametes to be obtained from it. Either capital S and small y smooth yellow, or small s and capital Y wrinkled yellow. And when these gametes are selfed in the F2, you are going to have 4 possible genotypes. Thus, even though all the seeds will be yellow, there will be segregation in the ratio of 3 smooth seeds to 1 wrinkled seed, because you have one of them that is small letters, which shows recessive trait wrinkled, but the other 3 each has 1 capital S, which shows smooth, but all have capital Y, which shows yellow. So you have 3 smooth yellow and 1 wrinkled yellow being obtained at the end of the day. Now for the test cross, it is a cross between the individual of unknown genotype which you are looking for. You want to identify that genotype, with another individual that is homozygous recessive at both loci, on both chromosomes. Let's assume there's an individual with smooth yellow seeds with a genotype capital S, small y. Then for parental cross, you're going to have that genotype capital S, small y, being crossed with the homozygous recessive. Take note of this, the homozygous recessive. For a test cross, it's a cross between an individual of unknown genotype with the homozygous recessive. You can see they are all small letters, wrinkled and green. And this will give you only one possible gamete, SY. While the genotype you are trying to find out, the individual you are trying to find out the unknown genotype has capital S, capital Y, small y. Now when crossed, this is a Punnett square. You're going to have two genotypes, smooth and yellow and smooth and green, in the ratio 1:1. It is smooth and yellow because you have capital S and capital Y. It is smooth and green because you have capital S and small y. But what happens if the genotype is capital S, small s, capital Y, small y? We're going to see something different. Again, the homozygous recessive trait genotype is this. Always have that behind your mind that for a test cross, the cross between an individual of a unknown genotype with the homozygous recessive parent. Now, unlike the previous selfing, where we had two possible gametes, from this genotype we have four possible gametes: smooth and yellow, smooth and green, wrinkled and yellow, wrinkled and green. And when crossed with the homozygous recessive gametes, wrinkled and green, we're going to have four possible gametes: smooth and yellow, smooth and green, wrinkled and yellow, and wrinkled and green. So it gives a segregation ratio of smooth yellow, smooth green, wrinkled yellow, wrinkled green in the ratio 1:1:1:1. That's a test cross. For test cross, it usually gives you ratio 1:1, not a 3:1 ratio. And that confirms the genotype of the unknown individual. In summary, Mendel used the garden pea Piston sativum as experimental material due to its many advantages in genetic investigations. The presence of many sharply defined characteristics in this plant formed the basis of his experiments. It was easy to cross it, and it had discrete traits that may be studied: smooth and yellow and wrinkled seeds, yellow and green seeds, yellow and green pods, purple and white flowers. So it was easy for him to carry out his experiment. He observed that when a pair of contrasting characteristics is considered in a cross, one of the characters marks or covers the appearance of the other in the F1. And of course, that's the dominant character or trait. And when the F1 is allowed to self-pollinate, both contrasting characteristics appear in the F2 in a definite proportion of 3 of the dominant trait and 1 of the recessive trait. When two pairs of contrasting characters are considered together in a cross, the segregation of one character does not affect the segregation of the other. as long as both contrasting characters are located on different chromosomes. They will sort themselves out independently. Now, the results obtained in both cases are not affected by which plant is a male or female parent, because he made reciprocal crosses and found out that they both had equal contribution of gametes. And these findings led to the establishment of the first and second law of inheritance. Take note of these key points as regards to Mendel's law. He proposed the laws of inheritance after conducting two types of experiments on pea plants for 7 years. You have a monohybrid cross experiment involving a pair of contrasting characters and a dihybrid cross experiment involving two pairs of contrasting characters. The laws of inheritance include the law of dominance, which states that the dominant trait will be expressed in the first filial generation. The law of segregation, that the alleles will segregate and only one allele, and not both of them, will move into the gametes. And the law of independent assortment, that when you're looking at two pair of contrasting characters, that a pair of contrasting character assorts itself independently of the other pair. And law of independent assortment says that the inheritance of one pair of genes is not dependent on the inheritance of the second pair of genes. So far, we've looked at inheritance involving a pair of contrasting character, monohybrid cross, and two pairs of contrasting character, dihybrid cross. You could also consider a trihybrid cross, having three pairs of alleles. Assuming we make a cross between two true breeding pea plants, one of the parents having smooth yellow seeds and purple flowers, and the other parent having wrinkled green seeds and white flowers. It is expected that in the first filial generation, it will be heterozygous at the three loci, but the dominant phenotype will be expressed. It will be smooth yellow purple. That is smooth, because you have capital S being dominant to small s wrinkled. Yellow, you have capital Y being dominant to small y, which is green. And purple, because capital V be dominant over small v, which is white. Since there are three loci, the number of gametes that the F1 individual will produce is given by the formula 2 raised to power n, when n is the number of loci or allele pairs. In this case, we're looking at a trihybrid cross, three pair of alleles. So the possible number of gametes to be produced in the F1 will be 8. Now the number of allele pairs and the type of gametes and protein that can be produced increases and becomes more complex, as we can see in the table above. For number of types of F1 gametes, for two pairs of alleles, we have 4 possible gametes. For 3 pairs, 8. For 4 pairs, tetrahybrid cross, you're going to have 16. And for 5 pentahybrid cross, we're going to have 32 possible gametes. So the more the allelic pair, the more the gametes going to produce. And we also notice that the number of phenotypes to be produced in the second filial generation corresponds to the number of types of gametes produced in the F1 generation. But for the number of genotypes to be produced in the F2 generation, it's going to be 9, 27, 81, and 243. And the possible gametes combinations is going to be 16, 64, 256, 1024. So as the number of alleles increases, the possible gamete combination increases as well. This was a monohybrid cross, this was a dihybrid cross, as we saw in the Punnett square of Mendel. But for a trihybrid cross, you're going to have 64 possible garment combinations in the Punnett square. Now let's look at some genetic terms. By pure line, it means a population that breeds true for a particular trait. I said earlier that Gregor Mendel used pure lines for his experiments. Those are lines that spread true from one generation to the next. They always give the same traits. Phenotype is the outward physical appearance of a particular trait. The genotype is a specific allelic combination for a certain gene or set of genes, or the genetic constitution or composition of an organism. Allele, one alternative form of a given pair of gene. It could be tall or dwarf. In case of height for pea plant. We talk about allelic pair, a combination of two alleles, as you have here. You also have multiple alleles. More than two alleles exist for any specific gene, but only two of them will be found within any individual. You have the dominant allele is the allele that expresses itself at the expense of the alternate allele. The phenotype, which is expressed in the F1 generation from the cause of two pure lines. On the other hand, the recessive allele is an allele whose expression is suppressed in the presence of a dominant allele. The phenotype that it disappears in the F1 generation from the cross of two pure lines and reappears in the second filial generation. Homozygote, an individual which contains only one allele at the allelic pair. For example, it may be capital D, homozygous dominant, or small d, homozygous recessive. Pure lines are homozygous for the gene of interest. On the other hand, in the case of heterozygote, it's an individual that contains two different alleles at the allelic pair. For example, capital D, capital T, small t are heterozygotes. We have two different types of allele at the allelic pair. The monohybrid cross is the cross between parents at a single allelic pair. Usually AA capital crossed with AA small letters, which are recessive. The monohybrid is the offspring of the two parents that are homozygous for alternate alleles of a gene, which is this or this. Dominance refers to the ability of one allele to express itself at the expense of alternate allele. That means this phenotype we show in the first filial generation, while the alternate allele is maxed and appears in the second filial generation. And that allele, of course, is the recessiveness. Backcross. Backcross is the cross of an F1 hybrid to one of the homozygous parents. For pea plants, height, the cross would be capital T multiplied with capital T small t, which is tall, crossed with capital T, which is tall, or capital T small t, which is heterozygous tall, crossed with small t, which is homozygous recessive, homozygous short or dwarf. Although the backcross is a cross to fully recessive parent or recurrent parent, as the case may be. This differs from test cross, which is a cross between an individual of unknown genotype with the homozygous recessive parent. And hybridization is a mating or crossing of two varieties. And these varieties have contrasting characters, as in the case of the experiment of Mendel. I believe that this lecture, at the end of this lecture, you have learned a lot about the foundation of genetics and also the works of Gregor Mendel. Thus, I have some learning activities for you to refresh your memory to know if you actually followed this lecture and you have learned from it. If you are able to go through these learning activities, then it means that you've understood perfectly the lecture of today. If you were to define genetics and tell me the importance of genetics to life processes, list at least 5 branches of genetics. You were to tell me why Gregor Mendel regarded as the father of genetics. Discuss it. Try that on your own. Sit down, take a paper and biro, and try to answer these questions. If you're able to answer them, then you've gotten a full grasp and understanding of this lecture. What were the two experiments he conducted that led to the laws of inheritance? What were his major findings on inheritance of genetic traits as regards Garden P, Pisum sativum? What are the 7 discrete traits studied by Gregor Mendel? State his laws of segregation and independent assortment. What do you understand by pure lines, and why did he use pure lines for his experiment? What are the 2 ways for testing a hybrid genotype? You'll be able to differentiate a test cross from a back cross using relevant genetic symbols. And in a trihybrid cross, you're able to give the possible number of gametes to be produced. I believe that this lecture will be meaningful to you. You can go through it over and over again and understand it better. If there are any questions on this, you can raise it up on the LMS platform for further explanation.